Hi guys, I'm Jonathan Huang. I'm doing this video proposal for the course Computational Modeling of Cellular Systems, and I will be talking about the Wilson-Cowan model of neural population dynamics. So first, why do you have to study the collective behavior of neurons? In animals, the sensory information is introduced into the nervous system in the form of a very large-scale spatial temporal activity. So for example, this complex neuronal system shown in the diagram here. This is a mouse olfactory system which maps a lot of different olfactory information into a very tiny space of neural processing system. And here the Wilson-Cowan model describes the dynamics between two types of neural subpopulations, and it's also the one of first studies to treat inhibition as arising from exclusively inhibitory neurons. And this gives us a model with two interacting subpopulations instead of just one, as in the previous older models. So the model here consists of two different variables called E of t and I of t, and which represent the excitatory and the inhibitory neuron firing rates. And here we're assuming that the cells comprising the populations are very close in proximity, which means we can ignore the spatial interactions between different neurons. And they're also subject to very intense random interactions. Also, we know that all nervous processes of any complexity will depend only on the interaction between these two types of neuronal cells. This, along with some of the physics, will gives us the pair of integral differential equations as shown below, which writes out the E of t plus tau and the I plus t of tau in terms of the fraction of excitatory and inhibitory cells, and also the response function S of x. And we'll also be coarse-graining our model to turn the integral differential equation into just a differential equation. And the coarse-graining idea is basically mapping a high-dimensional system into a low-dimensional but faithful representation. And it's also everywhere. So for example, ball and stick model in organic chemistry, renormalization in QFT, and also proteome partition. And finally, we'll do the model extension by proposing to add in the Tsodyks-Markram ST-SP model of short-term synaptic plasticity for the excitatory outputs. And here we'll have a six-dimensional system by considering also the depression and facilitation of our model system, which will be able to do this because of the new large-scale simulation abilities of modern computational softwares. And finally, this is our page of references for this